Hi, I'm going to tell you about an approach we recently tried for discovering novel gene combinations for multi-targeted cancer treatment here at Systems Oncology. With the discovery of synthetic lethal interactions, there has been increased effort to find gene combinations that do not affect cell viability in isolation, but do when in combination. Such combinations would enable greater specificity in targeting cancer cells while leaving healthy cells unaffected. Cellular fitness can be affected by altered expression in more than 2 genes at a time, but studying these with traditional methods has been intractable in human cell lines at this time. So with this in mind, we thought to use a computational approach combined with data from a yeast screen. We based our approach on a paper by Zhang et al. That uses yeast data from a trigenic interaction screen done by Kuzmin et al. You can see the references for both of those citations. We mapped the genes these yeast genes to human genes using orthology data from Biomart. This gave us not only 1-to-1 mappings, as you can see here in yellow, but also 1-to-many mappings and a few many-to-one and many-to-many mappings as well. And these persisted even after applying a threshold to the orthology confidence measures. And here's an illustration of how that might look for a given yeast triplet, with 1 gene that maps to 1 human gene, and then 2 yeast genes that map to more than 1 human gene. So this gives us 3 times 2 times 1, 6 human gene triplets, all of which are assigned the same interaction score indicated by the shade of the triangle. So in the interest of time, please read the poster itself and the associated references for more detail. But building on the original method, we used not only protein-protein interaction networks from string, but also co-essentiality and co-expression networks from DepMap and HumanNet. The node embedding steps capture the topology of the digenic feature network so that we can extend that information beyond the digenic context and embed it with the original genes. And the meta-embedding integrates these features together. For the training set, the gene node embeddings are combined with the measured interaction scores from the original yeast screen. And then these are used to train the model or tune the weights of the various layers. The model can then be used to predict new interaction scores from the node embedding data alone. So here's an example with a triplet of three genes with their node embeddings. These pass through 2 separate layers. There's a fully connected layer, which generates a static embedding, and this multi-head attention with re0 module that gives us dynamic embeddings. And while the static embeddings are specific to the individual genes themselves, these dynamic embeddings are specific to the trigenic context. And then these are combined by taking the element-wise power of the differences between them, and then that output is passed through a fully connected layer to generate the final predicted interaction score. As you can see here, when we looked at the model predictions versus the measured yeast screen-derived interaction scores in our validation set, we found weak correlation with a Pearson rho of just 0.37. Out of curiosity, we did apply our model to the larger prediction set. And as you can see here, the 5 most high-scoring triplets that were from a subset we deemed cancer relevant because they contained either a tumor suppressor or an oncogenic driver gene, the scores appear to be driven by the presence of a couple genes in the triplet. This is likely due to some of the 1-to-many mappings in our set. And of course, the weak correlation indicates that our model does not generalize and we have high uncertainty in our interaction score predictions, so we didn't pursue these interactions further. Availability of appropriate training data is a challenge in AI-driven drug target discovery. We are always trying new ideas to see if we might be able to extend data that we already have in new ways. Unfortunately, it did not turn out as we hoped in this case. However, the hypergraph self-attention neural network methodology and feature embedding in Zhang et al. May still prove useful in this area with different data. Thank you.